Next up, we have Eric from Karamedica, I think another UCR company.
So I'm Eric Gosink with Karamedica, and I'm here today to talk about Chitovec, which is a nose-to-brain gene therapy product we've developed. Now, therapeutics for the brain are a big issue in pharmacology, and it affects nearly every family, some neurological issue. You would think that with a market like that Big Pharma would be heavily invested in such programs. But the truth is, and over the past decade, Big Pharma has been pulling back on their early neurology research programs. And the simple reason for that is that there's a very low approval rate for early stage research, and that's especially true for therapeutics in the neurology field. And the reason for that is that the brain capillaries are actually very tightly sealed. It's what's known as the blood-brain barrier. Molecules that may be in the circulation simply aren't getting out to the brain cells where they're going to have their effect. And so people have been looking at other ways to get molecules or compounds through. Gene therapy is one approach. Genes are often delivered using viral vectors, and as the discussion was just going on, viruses elicit a damaging immune response, so you're often limited in how you can dose them or escalate the therapy. So with our background in medical polymers and neuroanatomy, we were looking at what else could be used for delivering compounds to the brain. And that focused us on the use of chitosan, which is a natural polymer derived from crustacean cells. And it's a well-tolerated material that's been known for years, but we've ground it up with proprietary formulations into these nanoparticles that we're calling Chitovec, and that it allows us to deliver compounds to a number of different cell types. So we're envisioning this as a sort of a generic platform for gene delivery. And as our first proof of concept, we looked at the delivery of CD59, which is a complement inhibitor that inhibits inflammation. And we see, and this is something that is significant in many neurodegenerative conditions like Alzheimer's or Parkinson's. And the studies that we have shows that it's well tolerated, and that it's actually the mice that we've dosed have retained their memory in a model of Alzheimer's. So we're continuing studies to look at the pharmacokinetics and toxicology. We are needing to get some big partners to go into the clinics. Unfortunately, they've been saying that we really need non-human primates. So we're planning some small trials to do those primate studies to get us further along. Oops, whoa. But we're looking at, as a first approach to get into therapy, is using that product itself, the CD59, to look at these neuroinflammatory conditions, such as Alzheimer's, Parkinson's, traumatic brain injury. But the other indication would be any therapy where you want to reduce neuroinflammation, such as Biogen has its lecanemab antibody, but the trouble with using it is that it induces inflammation and bleeding in the brain. So we may be a supplemental therapy in that case. The final therapy would be looking at, or a final approach would be looking at licensing the Chitovec as a platform that would be an alternative for viral delivery of genes. And there obviously, as the therapies are going along in the clinical trials, the evaluations of the licensing deals could be potentially larger. For example, Voyager Therapeutics is licensing out for multi-millions of dollars its preclinical viral approach for delivery of genes. And the rates just go up from there. I think I'm out of time at this point, but this is our team with neuroanatomists and polymer chemists, and be happy to take some questions.
Great, thanks, Eric. Judges?
Thank you very much for the presentation. I studied the blood-brain barrier during my PhD, so this is a good reminder. The slide that you show on you see delivery to the olfactory cortex, how deep of the brain regions outside of the frontal olfactory cortex do you see?
It depends upon the formulation. We have a variety of different chemistries. I talked about these proprietary methods. Depending upon where you're, the size of the nanoparticles, the degree of acetylation that we treat them with, we'll see the tropism of the nanoparticles with their gene payloads to different areas. So we believe that what it's going in is on the olfactory and trigeminal nerves. So, but there is a fairly broad distribution across the cortical layers. I could bring in the science team to speak with you. I am not on the science team. So.
Thank you. And then my second question is, so this is the CD59 is an antibody. What's other cargo have you tried outside of?
We have been looking at EPO-E4 as well as some smaller therapeutic compounds. There are some other indications, but our focus has been with the CD59 because we actually started not specifically on Alzheimer's, but a subset, cerebral amyloid angiopathy, which there's a vascular bleed. You probably will know that there's a lot of vascular leakage going on in those conditions. So we were looking at treating that, but CAA is a comorbidity as Alzheimer's progresses.
Understood. Thank you.
Is this technology only for large molecules, or can it work with small molecules as well?
Right now, the formulation is set up so that there's an ionic attraction. Yes, if it was to be a small molecule, it might have to have some kind of cross-linker to hold it in place. Then, of course, how do you remove the cross-linker unless there's some, like esterases, perhaps? But we have not gone into that chemistry. There's just been simply a charge association between the genetic material payload that we're delivering and the chitosan nanoparticles. And
can you explain the mechanics of the delivery through the nose? Is it a spray? Is it
the hope is to become a spray.
Okay.
Right now, we're just using mice, and that's the state of the therapy that we are the application that we have. So that's a pipette or injection right up the nose. It's a question of how much is retained. We're doing some off-the-cuff dosing of how much volume you would need. But the thinking is it should be significantly less than half a mil with the concentrations of genes that we would expect to need to deliver to have the effect. All of this needs to be borne out, and that is probably another reason to look at non-human primates to get a larger dosing. We're thinking about going with the standard models of pigs or dogs, but the pharma parties that we've been speaking with have really been burned on anything but non-human primates, and so they're pushing us to at least take an approach there to see what sort of results we can get.
So it's not a physical distribution into the brain. These
no,
nanoparticles basically diffuse into...
Yeah, no, I'm sorry. So if I misunderstood what you were asking. Mm-hmm. The chitosan itself is cell permeant. Chitosan has been used topically for nearly a century. Its actions on tissues are well understood, but we also knew that it has a cell permeant ability. And so the exposure of the olfactory and trigeminal nerves gave us an early and easy access into the brain. And once it gets in, it can distribute through the cerebral spinal area, and it distributes across the cortex.
Two questions. For the non-human primate studies that you're planning on, what's the delivery method that was going to be used in those? Is it going to be more pipetting, or?
That remains yet to be done. This was a recent twist that we've come across. So just the scale up. The fact that we've got good results, repeatable good results with rodents was our latest major step forward. And as we're speaking with an FDA intercept planning session, as well as we started speaking with large pharmas to try and figure out how to best deliver it. There are a number of different atomizers that are out there, but the question is what sort of concentrations we're going to need, what sort of gene dosing we're going to need on the nanoparticles remains yet to be developed.
And in those rodent studies, did you also assess any safety or damage to the tissue at all, the nasal membranes?
I don't believe the nasal membrane has been looked at. Again, I'm not on the science team, So I may take it, I would, if you want to speak with them, I'd be glad to put you in touch with them. But I don't recall discussions of that occurring. The overall systemic effects on the mice, there seem to be no effect other than the fact that in this Alzheimer's model, the untreated ones obviously didn't respond well to a maze test, whereas the ones with the CD59 retain their memory of the maze.
Thank you.
What would be the risks from business perspective for this? Like in terms of like competition, who is the competition? What other technologies are that you are seeing as a risk?
Well, there are a lot of different therapies out there. You know, a lot of people are using viral vectors, and so that alone is one big thing that we have to show improved efficacy without any kind of systemic injury. But it may have fewer applications outside of the hard-to-reach tissues like the brain. Because if you use a lipid carrier, for example, that's fairly innocuous, and you can deliver that pretty much everywhere. It's those hard-to-reach tissues that we may be better suited to going after, and fortunately the brain is a very valuable space to be in, but it may be that in other spaces that there are better alternatives. We just haven't been there yet, but we're addressing the brain, the access to the brain issue.
And are you considering only staying within this technology and licensing it out, or also maybe expanding to your own molecules for particular targets? We came into this because of the neurosurgeons' connections with the progressions of Alzheimer's and the vascular breakdown. So that's what brought in the idea of looking at a complement cascade inhibitor. There are a number of complement cascade related diseases out there that we could have applications for. So our CD59, for example, that molecule alone could potentially have applications in many, many other diseases where there are only a few of them have actually have FDA-approved therapies out there on the market. So we could be an alternative for some of those, but like I said, there could be other carriers that could deliver those molecules. There's a whole other side to the company relating to larger structures of the chitosan for both as non-compressible hemorrhage blockade as well as drug delivery as well. So there's a variety of different applications. This was just one more application that we saw for the nanoparticles because they have that permeating ability to get through cells.
Any final questions? All right, well, thank you so much. Excellent.